Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Aviraj. Welcome to our Club 250 Matchmaker sessions. We are starting our biochemistry module, and in this video we will be discussing some basic theoretical part about carbohydrate, lipids and amino acids. So carbohydrate is a water carbon, and most of the formula CnH2O, where N is the number of carbons and M is the number of H2O. Monosaccharides, that is, they generally have the formula C6H12O6, contains examples like glucose, fructose, and galactose, and disaccharides. So when you combine two monosaccharides, then that is called as disaccharides. So they are broken down to monosaccharides in the GI tract. So lactose, which is broken down into galactose and glucose by the enzyme lactase, is one example of disaccharides. Also, sucrose, which is broken down by sucrase to fructose and glucose, is another example of disaccharides. So there are some complex carbohydrates like polysaccharides, which are polymers of monosaccharides. Starch is a plant polysaccharide which contains glucose polymers. Also, glycogen is an animal polysaccharide which also contains the glucose polymers. So cellulose is a plant polysaccharide of glucose molecules which have different bonds from starch, and they cannot be broken down by animals. That's why they are called the fiber. So whenever you take high fiber in diet, they contain a high amount of cellulose which cannot be broken down. So that will improve the bowel function and that will relieve the constipation of the patient. So glucose, so all carbohydrates are broken down into glucose, fructose, and galactose. And when the carbohydrate through a complex process, which we will discuss in another video, is broken down to glucose. Now this glucose can go into various pathways, including the anaerobic metabolism and can form lactate. They can go into TCA cycle and can form H2O and CO2. They can go to hexose monophosphate shunt and can form ribose and NADPH, and they can go into the fatty acid synthesis and can form fatty acids. They can go into the glycogenesis and can form glycogen. So glucose metabolism generally occurs in the liver, brain, muscle, or heart. So liver have the most varied use of glucose. There is TCA cycle for the ATP synthesis, and there is also glycogen synthesis happening in the glucose, happening in the liver. Brain, there is a constant use of glucose for the TCA cycle, and there is only a little glycogen storage in brain. Muscle or heart, there is TCA cycle, there is use of ATP, there is transport into the cells heavily influenced by insulin. So if there is more insulin, there will be more glucose uptake, and the muscle and heart also store glucose as glycogen molecules. In red blood cells, since there is no mitochondria, there is use of glucose for anaerobic metabolism. They generate lactate, and they also use glucose for the HMP shunt. The adipose tissue mostly converts the glucose to fatty acids like muscle, which uptake and the uptake is influenced by the insulin. So if there's more insulin, there will be more glucose uptake by the adipose tissue. So glucose interrupts cells. So there is sodium independent entry and there is sodium dependent entry. So there are 14 different transporters which are described for the sodium independent entry channels, which are from GLUT1 to GLUT14, and it varies by the tissue. For example, GLUT1 is in RBCs. So sodium dependent entry, when the glucose is absorbed from low to high concentration. So whenever the glucose is absorbed from different concentration without the use of sodium or sodium channels, then that is called as sodium independent entry. But whenever we use sodium to absorb the glucose from low to high concentration, then that is called as sodium dependent entry, and that is in the intestinal epithelium and renal tubules. So in the intestinal epithelium, so we can see there is the sodium glucose co-transporter. So whenever there is transport of glucose, whenever there is transport of sodium, glucose gets inward through the same channel. Okay, there is co-transporter of glucose. So there is sodium glucose co-transporter. And again, inside there is GLUT2, which is a glucose transporter 2, which only transports the glucose through that channel. Okay. Also in the proximal tubule, there is sodium and glucose co-transport. And again, from the glucose transporter, there is glucose transport through the interstitium or towards the interstitial blood. So glucose entry into the cell, for example, GLUT1, which is insulin independent, uptake when glucose is high, and it is found in the brain and RBCs. GLUT4 is insulin dependent, which is found in the fat tissue and skeletal muscle. GLUT2 is insulin independent, which is bidirectional. So remember, two for two direction. And it is found in the liver and kidney and intestine and pancreas. So lipids. So let's move towards some basic overview of lipids. So lipids are mostly carbon and hydrogen, and they are not soluble in water. There are many types of lipids like fatty acids, triglycerol, cholesterol, phospholipids, steroids, and glycolipids. So fatty acid and triglycerides. So most lipids degraded to fatty acids in intestine, and enterocyte converts this fatty acid into triacylglycerol, and the chylomicrons then carry through the plasma. The triacylglycerols are degraded back to the free fatty acids by lipoprotein lipase in the endothelial surface of capillaries, and they are abundant in the adipocytes and muscle tissue. So let's learn some sorry, vocabulary. So saturated fat or fatty acid, which contains no double bonds, are saturated with hydrogen. They are usually solid at room temperature and they raise the LDL cholesterol. The unsaturated fat, which contains at least one double bond, is monosaturated. So monosaturated means one double bond, and polyunsaturated means more than one double bond. So trans fat is when there is double bonds can be trans or cis. So most natural fats have a cis configuration, and trans from partial hydrogenation can increase the LDL and lower the HDL. Okay, so these things are not generally tested in the examination, but they are necessary to understand the different pathways and different mechanisms. You must have heard about the omega-3 fatty acids, okay, which are generally found in the fish and fish oil. So they are the type of polyunsaturated fatty acids, and they lower the triglyceride level. So fatty acid metabolism. So there is fatty acid synthesis, fatty acid storage, and there is fatty acid breakdown. So fatty acid synthesis occurs in the liver, mammary glands, and adipose tissue in small amount. The excess carbohydrates and proteins are converted into fatty acids. Fatty acid storage occurs into the adipose tissue, which are stored as triglycerides. The fatty acid breakdown is through beta oxidation, which we will discuss in another video on lipid metabolism. So amino acids, they are building blocks of proteins. They all contain amine group and carboxylic acid group. All except glycine have L and D configuration, and only L amino acids are used in human proteins. So histone contains the basic amino acids. Their high content of lysine and arginine are positively charged. Okay, so the content of histone is important. Lysine and arginine, maybe after in the examination. Histidine is also considered a basic amino acid. They have a side chain close to the plasma pH. In case of sickle cell anemia, there is exchange of polar glutamate for non-polar valine in hemoglobin protein. Now, this line is also very, very important, very frequently asked in the examination. So, proline is a rigid structure or ring formed from amino group and side chain, and they are used in collagen. So, there is glucosenic versus ketogenic amino acids. So, glucosenic amino acids can be converted to pyruvate or TCA cycle intermediates, and they can become a glucose via gluconeogenesis. The ketogenic amino acids convert to ketone bodies and acetyl-CoA, and they cannot become glucose. The most amino acids are either glucosenic or glucosenic and ketogenic. So amino acids, for example, tryptophan is formed from niacin, so serotonin, melatonin. Okay, now they can form that. Histidine, histamine, glycine. Okay, they can form the heme, arginine, and so and so. Homocysteine can cause homocystinuria. Cysteine can cause cystinuria. And branched amino acids causes maple syrup urine disease. So carcinoid syndrome, which is caused by GI tumors that secrete serotonin, is altered by tryptophan metabolism. Okay, so there is normally nearly 1% of tryptophan which is converted to serotonin, and up to 70% patients with carcinoid syndrome, there is tryptophan deficiency or pellagra reported. So there can be serotonin effects like diarrhea, increased fibroblast growth, which can cause valvular lesions and flossing in this patient. Talking about heart knob disease, which is the absence of amino acid transporter in proximal tubule, they are autosomal recessive, loss of tryptophan in the urine occurs, and the symptoms occur from the niacin deficiency. So in case of pellagra, there is hyperpigmented rash, exposed areas of skin, red tongue, diarrhea and vomiting, and they can also cause dementia. So we have to remember dermatitis, dementia, and diarrhea. The treatment is high protein diet and supplement of niacin. So branched amino acids are the essential amino acids, and they are primarily metabolized by muscle cells. Metabolism depends upon the alpha ketoacid dehydrogenase. So the branched alpha ketoacid dehydrogenase complex is similar to pyruvate dehydrogenase complex, and there are E1, E2, E3 subunits, and the cofactors for alpha ketoacid dehydrogenase are thiamine and lipoic acid. So the maple syrup urine disease is where there is deficiency of alpha ketoacid dehydrogenase is an autosomal recessive disease, and they have five phenotypes. So in case of classic, the classic Naval urine disease is the most common one. Okay, there is increased branched in amino acids and alpha ketone acids in plasma. The alpha ketone acids of isolation gives urine a sweet smell. There is neurotoxicity. There is primarily accumulation of leucine, and the patient complains of lethargy, irritability, apnea, seizures, and there are signs of cerebral edema. The diagnosis is mainly based upon the elevated branched and amino acid levels in the plasma, that is valine, leucine, and isoleucine. Very important. Please remember these terms: valine, leucine, and isoleucine. The treatment is by dietary restriction of branched and amino acids and thiamine supplementation. So 28-year-old woman presents to the emergency department with severe flank pain and hematuria. She reports a history of recurrent kidney stones. Lab studies revealed the presence of cysteine crystals in her urine. Given the patient's presentation and findings, which of the following is the most likely diagnosis? Renal cell carcinoma, nephrolithiasis, cystinuria, polycystic kidney disease, and acute pyelonephritis. So cystinuria is the most likely diagnosis in this patient. Cystinuria is an inherited disorder characterized by impaired reabsorption of cysteine and other dibasic amino acids in the proximal renal tubules, leading to the formation of cysteine kidney stones. The presentation of severe flank pain and hematuria, along with a history of recurrent kidney stones, is highly suggestive of cystinuria, and the presence of cysteine crystals in the urine further supports this diagnosis. So cystinuria, there's two cysteine molecules which are linked together. So in case of cysteine, there are two cysteine molecules which are linked together. So cystinuria is an autosomal recessive disorder where there is decreased reabsorption of cysteine by the proximal tubule of nephron. The main problem is kidney stones, and the prevention is from the methionine-free diet. So again, the hereditary defect of renal PCT and intestinal amino acid transporter that prevents the reabsorption of cysteine. Cysteine is made of two cysteines connected by disulfide bond. The treatment is by urinary alkalization and chelating agents like penicillamine and good hydration and diet low in methionine. Thank you for watching. Keep studying hard.